everyone. Welcome to 17 Minutes of Science. My name is Sarah Cheeseman. I'm a technical solution scientist here at InVivo Biosystems. I'm glad you're back with us today. We are doing some episodic special interviews with members of our team at InVivo Biosystems so we can showcase the wonderful people we have on our staff that do all kinds of interesting science. And I'm totally delighted to welcome Dr. Adam Saunders, who's a key member of our technical team here at InVivo Biosystems. I'm going to do a quick intro about Adam, and then I'll let him say a few words, and then we'll go into the questions as we always do, in sort of our speed dating format. So I will just let you know that Adam is a multi-talented person, and he actually began his academic studies as a musician at Indiana University, but he later pivoted into biology. And it was at that point he was introduced to the nematodes, the elegans, while working with Dr. Susan Strom and Dr. Bill Saxton. And then he later went on to earn a PhD at Stanford University with Dr. Phil Beachy. He's worked on a variety of different types of signaling mechanisms that are essential for animal development. He later did a postdoctoral research project with Dr. Victoria DeRose at the University of Oregon. And then we were fortunate enough in 2019 to have Adam join the team at InVivo Biosystems. So welcome, Adam. We'd love it if you'd say a few words, and then we'll go to the questions.
Thank you, Sarah, and thank you for the kind introduction. So as Sarah mentioned, I'm a scientist with InVivo Biosystems, and I've been here since 2019. And largely my role here involves interacting with the clients and translating their needs and their project ideas to our outstanding technical team, and then working with the technical team to execute those experiments and make sure that they're delivered both in a timely fashion and with good scientific integrity.
And you always do with the team. So we'll start off by, of course, talking about some worms. So can you tell us about why it is that C. elegans, as a microscopic worm, are so well suited for lifespan and healthspan studies? These, of course, are some of the core interests of the company. You've been involved with all of that. So to help orient our audience to why are they such a great model for this?
Mm-hmm. Yeah, and it really stems largely from what makes the nematode worm C. elegans such a powerful system for developmental studies. So back in the mid 20th century, Sidney Brenner and his colleagues, they had initiated this model system largely because it was a small, simple organism that could be cultivated easily in a lab with just simple microbiological tools. But what really came out of it was it had this perfectly choreographed developmental pattern in which every cell division, going from the single-celled egg to an adult hermaphrodite or male that has slightly under 1,000 total cells, every cell division could be perfectly mapped out with some exceptions. But as far as animal systems go, this was an outstanding tool for being able to track pathways of development. Now, where that falls into lifespan and health span studies is that because it has such a defined trajectory of its life cycle, going from that single cell to an adult to finally its death, it emerged as a very useful tool for the initial studies into lifespan and longevity. And what came out of that, and what really launched the longevity field back in the 1980s and the 1990s, was the discovery that by inhibiting specific genes, you could greatly extend the lifespan of these worms, or reduce the lifespan. But that brought out the paradigm that aging and lifespan were not just sort of an entropic degradation of cellular systems, but something that could be developmentally and genetically regulated. And since then, the studies in worms have greatly expanded and reinforced this, but also really launched the field of aging as a medical field in itself. And there's this link between aging and this plethora of age-related diseases, where the common factor is really the aging. And so the worm has remained the workhorse of that, and a benchmark for studying therapeutic interventions into aging and longevity.
That is a great synopsis, and extraordinary to think that an animal seemingly as simple as a worm can have produced data that is impactful when we think about human health. I think a lot of people might find that really surprising, but as you say, there's a huge body of literature supporting that, which leads me to one of the things that we have offered in recent times is what we call our longevity platform, which is based on exactly this lifespan and healthspan measurement. And you were a key person helping to develop that service offering that we have. Can you tell us more about that process and help orient our listeners to what that built upon?
Mm-hmm. Yeah, well, as I mentioned earlier, the physiology of the worm and the initial genetic studies into aging, if you combine that with also the compound studies and the therapeutic interventions, the majority of compounds that have been linked to extended lifespan in mammals and even in some cases humans do have the same effect in C. elegans. So there's a high level of predictivity and translatability. And so it's hard to say what the exact percentage is, but because of that utility, the worm is and other small animal models are in a position to help accelerate studies into longevity and aging or probing of various therapeutics, because you can take a pool of those potential therapeutics and either find candidates or eliminate candidates based on their efficacy or toxicity very, very early in the process without having to go through expensive and time-consuming mammalian studies. And so our development of the longevity platform was really based on addressing this need in both the pharmaceutical and the nutraceutical market for a rapid screening approach for compounds that could influence longevity. And our first priority in that was to develop a rapid, robust, and very reproducible system that could be applied at a relatively high throughput. And so we had a team assembled to develop this product, and there are many different ways of doing lifespan. So backing up a little bit, how do we study lifespan in worms? Well, the worms have this attribute where you can synchronize them, so they're all the same age down to the hour in proceeding through their life cycle. And basically, the worms would be put on a plate where they can live out their lives eating their bacterial food until death. And those deaths, traditionally those would be scored manually, but that introduces a lot of variability. And it's been shown that most of the variability in longevity studies often come from the observer or the operator. So we had to do something that was automated. Well, there are a number of automated systems out there, and we adapted something called the automated lifespan machine that was developed by Nick Strustrup and others that had also been adopted by something known as the Canariditis Intervention Testing Program. So this is a program that is funded by the NIH to standardize practices used for testing lifespan and therapeutic compounds in C. elegans. So this is a consortium of three institutes, the Buck Institute at the University of Washington, the University of Oregon, and at Rutgers, that have done both automated and manual lifespans and really set the parameters for what makes a valid lifespan assay. So we've taken our cue from what's published and what's really established as the best practice in the community to bring it into this platform that we can offer to the broader pharmaceutical and nutraceutical community. But anyway, as far as the process of building that, this is something that required a fair amount of infrastructure. So there was a small team that was collected that was dedicated just to getting this basal platform launched. And so they worked for a month just only doing that to get this particular device up and running. And that forms the basis of the platform, but that's not all of what the longevity platform is. So the longevity platform, in short, it's a modified scanner that takes pictures of worms, and then we use automated image tracking and detection as well as some machine learning software to determine the times of deaths for those worms. And so we can get a large number of worms, a large sample size, very statistically powerful data. But on top of that, a lot of therapeutic interventions out there, they might help worms or other animals live longer, but we want people living healthier lives, and we want to be able to measure what we call the health span. So if it's great if you live to 120, but how are those last 50 years going to go for you? What we want is, and what's really relevant to the healthcare field is helping reduce those costs of late life care. And so extending lifespan alone is not going to address that. So with this machine from the automated imaging, we also are able to measure health span. But the third component that really has added a lot and really given us a lot of insight is the mechanism of action. So we do a parallel transcriptomic analysis of young and aged worms and compare the gene expression profiles of those to understand when a client asks us to test their compounds, where is it acting? Is this something that's analogous to some of the established and published life-extending compounds such as rapamycin or resveratrol, or some of the things found in pomegranates or other natural extracts? Is it hitting the same pathways, or is it's something analogous to caloric restriction or the pathways that are affected when insulin signaling is disrupted, or any of the other genetic perturbations that were discovered early on in C. elegans. So it's really those three components: the lifespan, the health span, and the mechanism of action that really provide us some interesting biology. For us as scientists, seeing the basic biology that comes out of this has really been exciting.
And as you say, all of those elements of the platform together paint more of a full picture, which is when it really comes into 3 dimensions. It sounds like when looking at those sets of data. Let's switch gears for a second. So you've been at both in academic labs, you've now been in a company setting. Some of our audience may be wondering what it's like to work in a startup company. So as we wind down the clock here, give us your takeaways, your impressions, some of the things that surprise you about working in a startup environment.
Mm-hmm. Yeah, and I don't know if it surprises, because I mean, going into my academic training, I was down in sort of like the Silicon Valley area and in a hub of biotech, and so my eyes were definitely geared towards the startup and biotech community there. But I would say the things that I really enjoy about it are the need for flexibility, the need for adaptability, the need to play many roles. So some of the roles that are not in the job description, and you have to really step up. And particularly in a startup situation, not everyone's going to be a specialist. You have to be prepared to interact with the marketing team, interact with the sales team, interact with the upper management, and then go to the bench and help somebody with their some of their basic bench skills. So it's really fast pace, and you have to be ready to switch gears very quickly. But I think as far as people who might be trying to consider what kind of track or where they want to go, I would say that being in this situation really allows you to not only take advantage of a diverse range of skills that you may have, but it also places an emphasis on being able to recruit team members and bring in expertise from a wide range of fields in order to meet certain goals.
That's a great summary. Never the same day twice. So in our last couple minutes, let's return to the fact that you are a musician and enjoyed being in that realm for quite some time.
You're not asking me to play, right?
Well, I don't know. Did you bring an instrument?
Not today. Not today.
What have you got down there? I can't see.
I have a pair of spoons, maybe.
Okay. We'll pass on that. But do tell us your instruments, your favorite instruments, and then how you made that switch from a musical career to scientific.
Yeah, it was largely woodwinds. I went to Indiana on a music scholarship, and so saxophone, clarinet, bassoon, flute, those types of things. I did a wide range of things from jazz music to classical music to country to spiritual type of stuff. So into musicals, it was kind of everything. So a really wide range of things. I was really kind of geared towards being a professional musician at that point in time. But I'd grown up with a love for biology and a love for science, and I had many family members that were either medical professionals or farmers or scientists, and that really drove the conversations around the dinner table, so to speak. And really there's a realization, as I was progressing musically and having to take some time off for an injury, that why specialize? I had the tools I needed musically to enjoy it and continue practicing it. But there was, particularly in the wake of September 11th, there was kind of a revelation as far as, okay, well, what other ways can I develop? What other things can I contribute and bring to the table? So thought, why not add the science? Why not add the biology and add that as well?
Wow. That was exactly our timer when you wrapped up that thought. And I was just thinking as you were talking, exactly, why compartmentalize, right? Mm-hmm. We continue to grow and incorporate all the things we learn and love. And that's great to hear that you're still playing the spoons. Mm-hmm. So I want to thank you, Adam, for taking time out of the lab today to talk to us, to tell us about your journey, and really appreciate working with you. And so we will be back for another episode of 17 Minutes of Science with another one of our colleagues. But until then, thanks everyone, and we'll see you next time.
Right. Thank you so much, Sarah.